Thank you, Professor Croce. Last but definitely not least in the talks is Professor Bert Vogelstein. And again, I'm not going to describe all his awards, just to mention only one of them, the 2013 Breakthrough Prize in Life Sciences, but this is, again, only a partial list of the prizes and awards. Again, going back, I'd like to start with a very old paper by Bert Vogelstein, which actually I think this is still the most cited paper, at least one of them, actually describing a technique to label specific pieces of restriction digest pieces of DNA by nick translation that can be used as a probe, and all of us, many of us, used this technique for many years. And this is not, it's not surprising, because if you follow Vogelstein's career, over the years he was involved in the invention of many technologies, many genomic and genetic technologies that allow him and his team to go to ask the major questions in cancer. Another very known work from Vogelstein group is the demonstration over the years of the acquisition of mutations during colorectal cancer development. And you all know this scheme that is also known as Vogelogram, where the acquisition, the sequential acquisition of mutations during the transition from normal epithelium towards adenomas, towards carcinoma, towards metastatic disease. Actually, each mutation, either in oncogene or tumor suppressor gene, result in a small increase in cell cycle or in some increase in the rate of proliferation that over the years result in the emergence of the malignant clone. So for sure, Dr. Vogelstein's work actually transformed cancer research through the work on colorectal cancer, but in recent years on many other types of cancer. As I mentioned, they were able to show that cancer is the result of acquisition of sequential mutation, and this became a paradigm, the major paradigm for modern cancer research, and it has a lot of implications for both diagnosis and treatment of cancer. Dr. Vogelstein was involved in the identification of many tumor suppressor genes and oncogenes, among them the APC gene, which was shown to be the gatekeeper. Actually, the vast majority of colon cancer tumors must have mutation that in APC to allow development of the tumor. And their group also characterized many other genes relevant to cancer development and progression, just to mention the mismatch repair genes that are important for genetic stability. Based on this finding of the APC and the MMR genes, they were able to develop blood tests for diagnosis of a patient with inherited predisposition for colorectal cancer. Then another major finding was the realization that P53 is the most common gene that is mutated in human cancer, but also the demonstration that while the prevailed dogma in the early 80s was that P53, actually a very Israeli gene, cloned by Moshe Oren from the Weizmann Institute, so this gene was thought of as an oncogene until Bert Vogelstein group showed that actually this is a tumor suppressor gene. The Vogelstein group was the first to perform a genome-wide analysis of all the coding sequences in a variety of tumors. They were pioneering this, and actually they analyzed more types of tumors than any other group or laboratory. And these studies, with the many different similar studies that emerged following this approach, actually mapped the vast majority of genetic alteration in human cancer. So this enabled the Vogelstein group to decipher the main principle of cancer, the number of mutations, the type of mutation, the number of mutation in different type of tumors, the role of stem cell divisions in the development of cancer, the rules that discriminate between tumor suppressor genes and oncogenes, and many more examples. And finally, and I believe this is what we are going to hear about, in recent years, Vogelstein and his team are now trying to utilize this deep understanding of cancer for better diagnosis and better treatment of cancer. Please.
This one. Thank you for that very kind introduction. It is a privilege to participate in this symposium together with Carlo and Mary Claire. Let me start, I guess, with a story about several months ago. I was traveling to Washington, D.C. from my home in Baltimore through back roads, and I noticed this sign on the road. It said, "Talking dog for sale." And so, being a curious fellow, I stopped. I went into the trailer, and there was this gentleman watching TV, smoking a cigar. He had one of these shirts without sleeves, T-shirt, and I said, "Where's this dog?" And he said, "Well, he's in the back." So I go in the back, and there's this dog, and I say, "Well, what's your story?" So the dog tells me that when people found out he could talk, they immediately recruited him to the CIA. You know, it's the U.S. equivalent, I guess, of the Mossad. And he did a stint in Eastern Europe, and nobody suspected that a dog could understand or talk, so he helped catch a lot of bad guys, and he went to Iraq and Afghanistan and Iran. Then he said he's retired, and now he raises his own puppies. So I said, well, thank you for your service. So I go back inside and I say, "I'd really like to buy that dog. How much is it?" And the guy says, $10. I say, $10? That dog is amazing. Did you hear about all the things he's done? And the guy says, don't believe any of that. That dog's a liar. So now I bring this story to your attention because here's a man who could not see the forest for the trees. And it's relevant to cancer genetics because there's now so much information. Every week we are bombarded with new sequences. Literally tens of thousands of tumors have been sequenced in their entirety. Millions of somatic mutations have been observed. And it's confusing, even to those of us who have devoted their lives to studying cancer genetics. So what I'd like to do this morning is to try to cut through some of those trees to see the forest, the basic principles of cancer genetics, and in particular how these principles can be applied to understand and hopefully in the future minimize cancer deaths. So I'm going to divide my remarks into 4 parts. First, the number of driver genes. Second, the source of these mutations. Third, heterogeneity among driver gene mutations. And fourth, driver gene mutations and their potential role in earlier detection. So here's the landscape of a typical pancreatic cancer. Each of those little tires represents a mutation. There are about 50, and this is true of most solid tumors, pancreas, colon, breast. It doesn't matter, somewhere between 25 to 75 somatic mutations per tumor. Now, if you just look at the driver genes, there are only a few of them. In general, about 3. And the peaks, the height of these peaks indicate the prevalence of these mutations in pancreatic cancers. KRAS mutations, for example, occur in close to 100% of the tumors, whereas P53 mutations occur in about 75%. There are only 3, roughly, mutations, sometimes more. Tumors continue to evolve. And the rest of the mutations are passenger genes. These mutations are irrelevant to pathogenesis. Now, they're not irrelevant to patients because they form the targets for current immunotherapeutic approaches, in particular immune checkpoint inhibitors. But they're irrelevant to pathogenesis. And most of the papers today, the genomics papers, report on passenger gene mutations, and they can completely be ignored when you're thinking about pathogenesis. Last week, together with Rachel Karchin, a bioinformaticist, and her student, Colin Tokheim, we looked to see how many bona fide driver genes there were. There are about 250 in the human genome, about 1.2% of the total genes. Slightly more than half are tumor suppressors, and the others are oncogenes. And now that so many thousands of cancers have been sequenced, this is a pretty complete list. There aren't that many coding genes that functions as drivers in cancers. And in fact, the majority of these driver gene mutations occur in only about 30 genes. If we were to understand these 30 genes much better than we do now, I'm sure we could make greater inroads into alleviating the cancer burden sooner. These are the really key genes that affect most patients with most cancer types. The other point of importance is how many genes it takes in an individual cancer to get from a normal cell to that cancer. And the answer is 3 in general. Again, it takes 3. Some cancers can have more than that. Cancers continue to evolve. But 3 appears to be sufficient for most solid tumors. The first one initiates the disease. It starts a benign tumor, microscopic in size, and it remains that way until a second mutation occurs, which results in expansion, still benign, but to a size that can be seen at the macroscopic level. And finally, the last strike, third strike in baseball terms, promotes invasion. This changes a benign tumor to a malignant one, a tumor that can't invade through the basement membrane to one that can, and can then metastasize to other organs. We do not believe currently that metastasis requires any more mutations other than these 3. After the tumor cell acquires the ability to invade, metastasis appears to be a stochastic process that will eventually occur over time. And over a lot of time, cancers do not develop overnight. As a result of both molecular and clinical analysis, we can estimate that most solid tumors take 20 to 30 years to arise. That is 20 to 30 years from that first mutation to the last mutation and the subsequent development of metastatic disease. And that's an important timeline because it says there's a lot of time to intervene in the process in this intervening decades. Now, what is the source of these driver gene mutations? Historically, if you look on Wikipedia or the American Cancer Society website, you'll see that there are two sources of the mutations that occur in cancer: environment and heredity. Well, that's incomplete. And the degree of that incompleteness has been illustrated by a mathematician named Christian Tomasetti, who has emphasized the role of random mutations, called R mutations, in cancer. And the basis for this quantification is pretty easy to convey. If you do a kind of Gestalten experiment, a thought experiment, and suppose you were to look at the concentration of a compound, let's call it compound R, a chemical, in various normal tissues in millions of individuals at autopsy, you might find that compound R had a very low concentration in normal bone, but very high concentration in normal colon. And then, if you were to look at the incidence of these same cancers in this population, you'd find that there was a very high correlation between the concentration of compound R and the incidence of these cancers that was in direct proportion to that concentration. Very high correlation. And your conclusion would obviously be that perhaps compound R is related to the incidence of various cancers. If you were subsequently to find that compound R is a potent mutagen, then your conclusion would undoubtedly be solidified. Now, compound R then likely contributes to the variation in cancer risk across tissues, but in reality, what Christian found was that compound R is simply cell division itself. Every time a cell divides, mutations occur. In fact, about 5 mutations occur every time a cell divides. And this correlation between the number of stem cell divisions and the incidence of cancer holds throughout the world, not just in the United States, but Christian showed it held across all 69 countries that were registered, 4.8 billion people represented. Now, how do we relate this to cancer etiology and the mutations that actually occur in cancers? What proportion of the mutations are due to random mutations generated during cell division rather than other factors? Well, the insight that Christian had can be very simply depicted mathematically. Suppose it takes 3 mutations to get to a cancer. Then, if some substance increases the mutation rate by 2-fold, then that should increase the cancer incidence rate by 8-fold. And that's true for smoking, for example, and can be shown to be true for other known carcinogens. And based on this, Christian could estimate the fraction of mutations that were due to random factors, cell division, versus exogenous agents in various tumor types. In lung, for example, most mutations are due to cigarette smoke, as would be expected. In brain, in contrast, virtually all the mutations in brain tumors were estimated to come from these random replicative mutations R. In fact, in most cancers, all 3 mutations that are required to transform a normal cell to a cancer cell are due to R. Now, This work caused a lot of controversy, which surprised us because we thought it was kind of obvious that cells divide and mutations occur. And all that we did was quantify the importance of these random mutations. But in retrospect, maybe we shouldn't have been surprised at all of the attention in the lay press this caused, for two reasons. First, it's somewhat unsettling to many people that even if one follows all of the guidelines from the American Cancer Society, that is, to avoid smoking, to avoid sunlight, exercise, eat right, even if you're an Olympic decathlon athlete, you can still get cancer. That's unsettling. We're not in total control. And secondly, it's somewhat unsettling from a philosophical viewpoint that we're subject to these random stochastic factors. Einstein is famously quoted as saying, God does not play dice with the universe, his response to quantum theory. Now, if Einstein had trouble with the randomness of the universe, I guess it's quite reasonable that others in the population, all of us, can have problems with this concept, but it's true. But to me, its importance lies not in its philosophical or ecumenical implications, but in its practical implications. If these random mutations are going to occur, regardless of how well we eat and what we avoid, it's extremely important to develop secondary prevention measures to prevent cancer. Secondary prevention means detecting cancers after they have occurred, but at an early enough stage so that they can still be treated. Primary prevention is always best when it can be practiced. Always best. If you can eliminate most lung cancers by avoidance of cigarettes, that's the best way to reduce lung cancer deaths. But in other cancer types not so associated with the environment, the key to prevention, I believe, is secondary prevention, and I'll get to that at the end of my talk. This brings us to the third topic I'd like to discuss, which is heterogeneity among driver genes. Each tumor has its own set of driver genes. They're often related. They're often in the same genes. And tumor heterogeneity is an active area of research. It's a bit over-tutored because amongst driver genes, there's little intratumoral heterogeneity. But one form of tumor heterogeneity that is very important is the heterogeneity within individual metastatic lesions, because this heterogeneity is what is responsible for the development of resistance against therapies. And here's one notable example, which I'm sure most of you are familiar with. This is the treatment of a melanoma patient who has multiple lesions on his skin with a BRAF inhibitor. The tumors almost miraculously disappear within a few months, a true miracle. But almost as miraculously, unfortunately, they often reappear in exactly the same place just a few months after they disappear. Now, the basis of this drug resistance is by now well known, largely due to the work of mathematician Martin Nowak and his team at Harvard. And its fundamental basis is that there are hundreds to thousands of resistant cells present in each clinically evident metastatic lesion prior to therapy. And I emphasize this word, prior. These mutations that can confer resistance are already there prior to any therapy. And this is why drugs fail. It's certainly not hopeless. Mathematicians like Martin estimate that if one could develop two drugs for which the same mechanism of resistance cannot come into play, then many tumors could be cured. So far, two drugs to treat most patients have not yet been developed, but perhaps in the future they will be. But this brings us to another question, which I think is very relevant, is why we can cure mice. Some might say that, well, mouse models, they're really just models. They're not the same as human tumors. But I think the sophisticated mouse models developed particularly by mouse molecular geneticists really are pretty good mimics of cancer. And we can cure many of these mice. I, in fact, am very good at curing mice. If you are a mouse, particularly a mouse that I have administered a tumor to a few weeks earlier, come to me. I can give you hope. Unfortunately, I'm not so good with people. Now, the reason I think it's easier to cure mice is illustrated in this slide. This is a typical CT of a patient in a phase 3 study. You can see all of the metastases, and I've just superimposed a mouse on this CT. You can see that each individual metastatic lesion is larger than the mouse, and if you just look at the mouse tumor, it's literally orders of magnitude, hundreds or thousands of times larger than a typical mouse tumor used in a preclinical study. It's just the numbers game. The fewer cancer cells there are, the less likely that one of those cells will contain a mutation that makes it resistant to the drug that you throw at it. That, I believe, is the main reason why it's easier to cure mouse tumors. And how to cure cancer then? Turn people into mice. Now, I say that partly tongue-in-cheek because earlier detection of tumors when they're still small, when there's micrometastatic disease, is essentially turning people into mice from a cancer therapy vantage point. And this conclusion really is buttressed by a large amount of clinical data. For example, if you treat micrometastatic disease in colon cancer patients, which is what all adjuvant therapies do, right? That's adjuvant therapy for colon cancer. You're treating patients who don't have evident metastatic disease, but they have potential micrometastatic disease, and it's been shown by oncologists that you can cure about half of them, 47% to be more exact, by conventional chemotherapy. If you treat advanced cancers, colon cancers, once they become radiologically detectable, you cure 0% of these patients. So it goes from 47% curable to 0% just by tumor size. And even with the new and so exciting immunotherapies, the same concept holds. This is a paper from Nature last year where it was shown in melanomas that patients who had low tumor burdens could often be cured, or at least driven into prolonged remissions, with anti-PD-1 immune checkpoint therapies, whereas patients with high tumor burdens did not respond nearly as well. And even with CAR T cells, the same observation, low tumor burdens, much higher probability of therapeutic success. My conclusion is that early detection, perhaps it should be called earlier detection, does not have to be very early to make a substantial impact on the survival of cancer patients. And this brings us to the last topic I'll discuss, which is driver genes and earlier detection. And our group has long been involved in trying to develop tests for such earlier detection. And most recently, we borrowed a page from our oncologist colleagues. It's well known in oncology that combination chemotherapeutic agents work better than single agents. This is a general principle of therapy, period. It's true for HIV, it's true for tuberculosis, it's certainly true for cancer. And what our group has recently tried to do is to borrow this concept and not to look at a single analyte for earlier detection or diagnosis, but multiple analytes. And there are lots of them. There are somatic mutations, there are copy number changes in the genome, there's epigenetic changes of both DNA and protein, there's microRNAs that Carlo just described, there's proteins, there are metabolites, there are lots of possibilities, hardly any of which have been investigated in modern times. The markers that are now used for detection of cancer, actually there are hardly any that are approved, but they were discovered literally decades ago, almost a half century ago. This is Josh Cohn's, our group's most recent work, published a few months ago, using one of these multi-analyte approaches. Basically, Josh Cohn used selected regions of 16 genes to develop a blood test, less than 2,000 bases, which he showed could detect cancers, or at least were mutated in cancers. These are solid tumors, the cancers, not the blood, in 80% of them. Again, only a small number of genes really are mutated commonly in many cancers, plus 8 protein biomarkers. He developed a test called CancerSeek, which he applied to a couple of thousand individuals, roughly half with cancer and half normal controls. And he developed this test so that eventually it might be affordable, perhaps something like less than $500. And the performance of this test was reasonable. These are the 8 different tumor types which were evaluated in the study, and it was encouraging that in 5 of these tumor types, one could detect at least 2/3 of the patients with cancers by this relatively simple blood test. And in each of these cases, a significant fraction, again, at least 2/3 of the cancers could be detected. Another important aspect of this study was that Josh developed a machine learning technique to try and predict the cancer site, which was detected by this blood test. This is important because if you do a blood test and there's cancer, you don't necessarily know where the cancer is. This algorithm allows one to tell where that cancer might be if the test is positive. And for example, in colorectal cancers, the algorithmic approach allowed Josh to identify that the site of cancer was in the colon in about 84% of patients, and was in one of 2 sites, either the colon or one other site, in virtually every colon cancer patient. To follow up this study, we're now engaged in one called the DETECT study. It's been carried out by the Geisinger Health System. It just started a few months ago, but the idea was to evaluate somewhere between 10,000 to 50,000 normal individuals to determine the sensitivity and specificity and hopefully potential utility of this kind of test in an average risk population. Obviously, it will take much work to get this experimental test to the stage where it can be applied to people. But it's not the only multi-analyte test that can be used to help diagnose tumors early. There are a variety of bodily fluids which can be used in similar ways. For example, Yuxuan Wang showed earlier this year that she could use the DNA from pap smear fluids to diagnose uterine cancers, as well as some ovarian cancers. These are the same pap smear fluids so successfully used to evaluate HPV and to detect cervical cancers over the past several decades. The same DNA can be used to analyze mutations that occur in uterine or ovarian cancers. And a similar principle can be applied to the DNA from urine to detect bladder cancers, saliva, pancreatic cysts, fluids, etc. The last modality, though, that I'd like to emphasize is imaging, which in my view is the best way to diagnose cancers early if advanced imaging modalities can be invented. Now, the Dan David Prize this year, as you all know, was for the future of personalized medicine. So let's look at the future. Scientists are not really very good at predicting the future. We're too tethered to reality. The best predictors of the future are science fiction writers. So here's one of my favorites. And it comes from a movie called The Elysium. Maybe some of you have seen it. It's about a world of the wealthy that orbits around our planet. And they have this really neat instrument there. It's in every home. Now, that's pretty good. So is it science fiction? Yes. Look at this. This would have been considered science fiction 50 years ago, but if you go 2 blocks down the road, you'll see one of these MRI machines diagnosing all kinds of things, including cancer. What seemed like science fiction 50 years ago is no longer science fiction. That's the general principle of science. And I'd like to conclude with showing you some preliminary results of our group's entry into this world. We had a simple goal with pancreatic cancer. In the US, there are 50 million abdominal CT scans performed each year for a variety of conditions, not for pancreatic cancer diagnosis, for auto accident injuries, all kinds of injuries, nonspecific abdominal pain. And it's been estimated that 30% of the pancreatic cancers that are actually evident in these images at a relatively early stage of pancreatic cancer development are missed by radiologists and only detected later. So the goal of the study that I'll show you was simply to provide a tool to help radiologists detect these early incipient cancers. And the project is called Felix. It's led by one of the world's truly great pancreatic cancer radiologists, Elliot Fishman, and Alan Ewell. Alan Ewell is a computer scientist at Hopkins, one of whose claims to fame is that he helped develop driverless cars. And when we explained this project to him, he thought it had a lot in common with the algorithms that he developed that are now used for driverless cars. So here's the progress report. So far, Elliot's group has expertly annotated, that means manually drawn, the pancreas and other organs in about 1,300 normal people's CT scans and about 600 pancreatic cancer CT scans. So the input for the algorithm, for those of you who are used to thinking about artificial intelligence and machine learning, is this. Just CT scans, black and white images. They're 900 slices per patient. And after about 16 months of algorithmic development, here's what Alan gets. A few seconds after you input a CT image, a CT scan into the computer, out comes this deep network prediction of the pancreas in 3D. It's really rather remarkable. And in addition to the pancreas, you see all the other organs in the abdomen, the blood vessels, the intestines, the stomach, spleen, kidneys, etc. Now, most importantly, and most difficult, is you can start to see pancreatic cancers. You can see the radiologist's interpretation of the scan on one side and the computer's interpretation on the other side. The yellow is the normal pancreas and the orange is the pancreatic cancer. The sensitivity so far is about 95% for detecting cancers, but these are cancers of medium size, 2 to 3 centimeters in diameter. Alan hopes that with further development of the algorithms, his group can begin to see tumors that are much smaller. What of the future? Well, I'm optimistic. Now that the genetic basis of cancer has been so well worked out through the efforts of so many thousands of scientists throughout the world, I'm pretty confident that cancer deaths can be reduced by at least 3/4 over the next generation. But I think that will only be possible if enough resources and research are focused on earlier detection of cancers, as well as much as now devoted to treatment of advanced disease in order to make this prediction come true. And here I speak particularly to the young people in the audience who are our future. I have 2 recommendations. First, when you think about minimizing cancer deaths, think not only about curing advanced cancers, but in other ways to reduce deaths. Earlier detection, other prevention schemes, things that I can't imagine, but hopefully you can. And second, and perhaps most importantly, think a little bit less like a scientist and a little bit more like a science fiction writer. Thank you for your attention.